I just want to use her as a fun kind of so just as a start, we just started recording. We're going around urgent needs for all the projects, and then we'll dive into some diagnostics threads. So this is the plant team. Use Anupma as a bounce point. So she spends a ton of time with kids across India, and she teaches antibiotic resistance to kids in the hope of, and again, she's built an entire category of workshops around this idea. And you can see it's a conceptually difficult idea, but then of course, hand washing and other sets of factors and kids taking medication and what does that mean? And then she does that through microscopy. So I want maybe a week in when you've thought about what it looks like so that we don't just keep diving into the technical bit of the detail, zoom out of it and see, pretend that she's the teacher, you are running a training for her, and what materials would go, and use that her as a bounce point and take a little bit of feedback saying if you were and I mean, she's remarkable with kids. So she's a very easy person. Like then we will choose somebody that's more average, who's tied up with teaching and just exhausted and is like, I don't want to talk about this one more thing. We will use that, but like you first want to start with somebody who's going to be over enthusiastic. So schedule a call, send messages on Discords. There are a couple other teachers in the community itself, so you can just send a Discord message saying anybody who is actively taught or has interest in teaching, we want to mock, because some of this will be about the object that you build. Some of it will also be about the materials around it. Okay, any other urgent needs? We'll do a design session, so we'll kind of bring up that in a second. Laser safety is important to think about with kids, if it's going to be lasers. So, yeah. What age group? Yeah. Okay. Okay, so, and I hear from Shrishti Sharma that although she's not part of the team, she'd be happy to brainstorm. I'm assuming, Shrishti, you're mentioning about the plant project that we're just discussing.
So I joined a bit late, so I'm not, I don't have complete
so
context.
Shrishti, I will do this for everybody who's not part of a team. I'll just talk a little bit through and have you guys join. It's important to join projects at this point. So just wait till the end of the class.
Okay, sure.
Yeah.
Thank you.
Okay, who wants to go next?
We can go next.
Okay.
So who
are the team members?
Passive ocean monitoring. We have Divya and Roy. Roy should be online as well.
Okay.
Yeah. And so I'm not sure if you saw the video, but we're kind of shifting focus, and instead of building like a CO2 sensor, we're gonna build a platform.
That's perfect.
Ocean floating platform that like module that other people can integrate their sensors into. And so like as our project delivery, we want to build this pretty much this floating buoy with power and communication. So right now we met and we're planning, and right now we need to model.
And it's 3 people, right? Yeah.
3 people, yeah. We're still modeling, let's say, like the actual structure and then the electronics requirements.
And so then just as a background on this, have you guys seen what SOFAR did? You guys know SOFAR? Okay, so let me just do a quick, I think when I had done it last time, I didn't do that in a, okay, can you guys see my screen? So this is called the spotter buoy. And SOFAR is a company locally, and we've been collaborating with them for a while. They started their origins in kind of open source threads. What's useful about it is it provides power, and it's being used for wave height measurements. So there's lots of things inside. Okay. So what would be valuable is we are anyway getting a SOFAR buoy for one of our projects. And there is a couple of SOFAR buoys also in Steve Monosmith's labs per se. So the first thing you guys should do is just look at it.
Yeah.
Look at it carefully. Super expensive right now, but their intents are still in the same space. they have solved a certain set of the solar panel problem.
Okay.
So far as a platform is not designed to mount things on top as yet. They have a lot of things that you can dangle on. So they have a power cord that then goes down, and you can implement lots of things on the bottom. But take a look. So you can see these sets of connectors, get a sense for what is there, and potentially
so and it's anchored on the floor.
You can anchor it, you can float it. It's quite modular in certain ways. And what they've been using them is as floating buoys for wave height measurements. That's one, but now this has grown significantly. But what I'm just trying to say is, look at this design explicitly. It's still good to build another version of it, but with a very cost-specific thing in mind that, oh, if this is, I think I'm remembering this is like 5K or something right now. And you might say, oh, that's too much. Or you might look at it as like, oh, I want to borrow this and this from this design, while our goal is really floating sensors and thinking a lot about a chassis, for example, in which the sensor goes in and a SOFAR buoy plugs in. So there are many ways of either using this hardware or expanding it to a point where it can actually be used as a sensing platforms for the air. Currently, they are wave height measurement devices, and they are massively used by the military to predict and map some of the physical oceans it's a low-cost way of measuring the height of the ocean. When you measure height of the ocean, you can tell a lot. So that's one. We should urgently get you access to one of these. So we can, after this class, we can walk to Steve Monosmith's space. I can at least show you one. I know a couple other students in this current batch are using. We are using it for Pelagica to begin with itself. So I want to make sure that we exploit as much of the knowledge that's been built in it. And read the history of this project, because there's a massive amount of work that they did in the open domain before they became a big successful company. So it's kind of, you want to use them as a launch pad.
Of course.
Okay, which sensor, if you were designing something, would be the CO2 sensor?
Yeah, surface CO2.
Yeah, okay. And in some sense, is there a replica, or is that sensor already working?
So the thing is, it's working, but it's not compact at all.
Okay, yeah. So because you cannot do it in an infinite, you need a mirror to come back.
They were measuring methane, and they said that CO2 is a much stronger absorption. Signal.
But you still need a mirror to come down.
So you still need a cavity. And they had one that they had the small, which mounts meters, they had 3 meters, and that was enough for CO2.
Okay.
So
3 meters.
Yeah.
3 meters folded.
Yeah.
Folded.
Yeah, folded.
So how big is your platform total?
The platform's going to be small. Sorry, the actual cavity is this small. The
optical path is 3 meters. I see. Okay. Yeah, 3 meters is, and it needs to be water protected. If water ever got in the cavity, you're dead.
And there needs to be some high-speed electronics as well, like a lock-in amplifier.
I see. Okay, and then that's what they're working on.
But that's what we're working on. It doesn't make sense to work on.
No, I think the idea would be is to build that as a platform. And even if you did, it would be perfectly valuable for the world if we bought a SOFAR buoy and you guys did the integration for that sensor for SOFAR buoy. As a team, that would be a spectacular project, because this would be the first time suddenly we can actually deploy. And then we focus on thinking a lot more about what is lacking in the current platform. So it's perfectly okay, because this type of an integration, using any kind of a sensor, like you can literally take a sulfur sensor, for example, that's commercially available, take the SOFAR buoy and see whether you can get data out of it, because it already has cell phone connectivity.
Okay, that's great. It wasn't aware.
Yeah.
So
what I'm trying to say is, it's perfectly okay to pivot when you're thinking about certain sets of things, because the end result is, can we create the vision of what you're thinking about, which is a large number of sensors that are on these platforms. And then the moment you try to implement one, you will see the missing pieces of the puzzle. You will see that, this platform is not actually viable. And because we were going to buy one anyway for Pelagica, so it's just useful, and for a lot of our work, might as well actually get access. And then for your team, how many, you're the only one local?
Yes.
Okay, and the other two people are where?
Are in India.
Okay,
Divya and Roy.
so one thing I'm gonna do, Divya and Roy,
Yes.
is I know the so far people, and what would just be fun, and this is open for anybody. They're in San Francisco on any of the weekends. I can organize a quick short trip. Just to see, oh, how does a company that makes these types of things operate and work? And you will see their fab facility, you'll kind of see their, it's just useful to get a sense of, oh, I can now see the path and the trajectory from the platform to an entity to now this fully functional thing. So we should, for you guys, we should plan a trip to SoFar.
Okay.
Yeah. Okay, let's keep going. Who wants to go next? Yes.
So my group is Aiden and Matthew.
Yeah.
They're both here.
Okay, and they're not in the room
They're
yet?
not in the
Okay,
room.
yeah.
We
Yeah.
diagnosis
Mhm.
device, and so we know that like one of the existing devices is like the transcutaneous emission,
Mhm.
but like because of skin pigmentation, it's not so
Yeah, it
effective.
doesn't work. So you reminded me something very important. I don't know how many of you know the crisis that happened during COVID about the pulse ox is not working for certain skin color. You guys know the story? So the FDA retracted a large number of pulse ox devices because darker. Sorry, pulse ox is how you measure your oxygen. So if you have COVID and your pulse ox reading goes below 95, go to the hospital. If it goes below 90 saturation percentage, you need to be in the emergency. So it was a very important triage device around the world. People were shipping pulse oxes were being used, like literally, people had them on them just to figure out. And then later on, published data showed that skin color, none of the original FDA approvals actually accounted for the broad populations. And hence they were not reliable in darker skin colors.
Were they overestimating or underestimating?
It was very dangerous, basically. So they were, to a certain extent, it was a key triage device because you were saying you are okay and you need to be ventilated. So it was a life and death decision. Even at the front gates of the hospital, people were turning people back. Like, come back if you are really below this saturation. And they were giving them a pulse ox. So what you're saying about skin, it matters. And it just, in the old days, of course, for many FDA approvals, you didn't need gender data. You said, oh, all data collected in males and was sufficient. And that has changed. So the FDA rules have changed. And now, at least for devices that operate literally on optical properties of skin, you would think that would have been, but it was a massive oversight. Okay, yeah.
Yeah, so we wanted to make like a frugal version of like a bilirubin.
Yeah, bilirubin, yeah.
And well, so what we were thinking was like just to bypass the skin, we would try to work with like a prick of blood. So then everyone is the same. And so we wanted to see if we could like use the light from the sun to look at the transmittance through blood. And then I think our project is basically thinking about the optics of how we can do that. So I think one idea we were having was so like there's this thing called light therapy where they shine blue light and that helps break down bilirubin. So we would like filter out blue light, see what wavelengths we're getting, like probably yellow, and then use some kind of photosensitive paper and maybe like compare it against a reference so it becomes clear to say like, oh, this sample is above a certain.
So already with the ideas that I'm hearing, I can see you're really thinking about an extremely low cost approach to this, right? So let's even assume no electronics, no power, no electricity.
Yeah, I think we were trying to say low power, but then if possible, it'd be nice if you didn't need any.
No, start with the most ambitious thing. What's the worst that can happen? You can fail. So photo paper, the sun, and a single lancet for a drop of blood, for example. So what's the most urgent things you guys need right away? First of all, you need permission to work with blood.
Yeah.
So we have those IRBs in the lab. Blood is tricky. Does anybody know why working with blood is tricky?
Bloodborne pathogens.
That's correct. But viruses,
Hmm.
no. HIV, for example, it's very easy. And so don't trick yourself until we've done the kind of paperwork. We have artificial blood in the lab. We also have access to sheep blood and others. We're testing certain sets of ideas. Because what you want to do is figure out, can you build a mock or a phantom in a non-human context before transition? So those would be 2 or 3 immediate sets of things. The other factor to think a little bit about is do some calculations first, because it's known what concentrations exist. How much transmission depth do you need for it to be detectable? Like, you need a tiny 1 millimeter drop, and that's sufficient? Because you could argue that if whatever your probe, your light is not sensitive enough, you might not be able to detect it. That small like transmission path.
If it's too thin.
If it's too thin, so then you might say, oh, I need 10 millimeter path, but then it's a long blood, then it's a capillary that you're talking about. So think through a little bit about these sets of geometries before you even think about a detector per se.
Okay.
I think the other part of this is, which age group are you thinking about?
We're thinking about infant jaundice.
Yeah. So then infant jaundice, you cannot take that much blood also. There's a limit to, it's usually a heel prick. So it's done on the heel of the kid, and it's usually a small amount.
You know, about
Yeah, usually a couple microliters. I think, I wouldn't go above like 10 microliters. No, 10 is too small, I would say. Usually when we do a glass smear, we do roughly between a single drop can be
Few hundred microliters to
what were we doing on our finger? Is that 100 microliters?
about. So
Intuitively, you're right. The number that keeps coming in my head is 20 microliter is a smear on a blood slide. Actually, yeah, 20 microliter is the thin film of a smear on a glass slide. So 100 microliter is a good number. One important alternative to light would be chemistry.
here's the paper-based that the SynBio people work on, like where they print all the assays on it already, and it's just kind of like water activated.
you just put the blood on.
Yeah, but what I'm just saying is compare optics to chemistry. Like, we should still pursue optics in that space, but really compare. At the end of the day, you want to make sure, start by making a device that can measure transmission properties of blood. Like, build that as a test case, because I want you guys to build something and fail early. So build a minimum version of what you can detect. And now I'm kind of going back and forth on, and I'm realizing the optical mouse has a laser source, it has a lens, it also actually has a really nice detector. And I'm curious, if you can hack an optical mouse, what would you get out as a test case? Just playing around with certain sets of things where you get numbers out, or build another version of it, which is what would be the minimal. You're not trying to take an image, you're just measuring transmission.
Yeah.
And there is a classic tool called fluorometer. And fluorometer are super expensive. And so thinking about what is a minimum object that measures transmission, but also in a very small scale, and allows you to get explicit quantitative numbers out. Like the moment you already put a camera, so I think Raspberry Pi would be the place to start. Even if you are thinking about ways of doing things without electricity, I'm just saying in terms of your setup, build a system, because PyCam is super, super easy. You get all the libraries for free. You'll start getting numbers out of it. And it's a good platform to know. But the key is you have to be quantitative about your readouts early on for them to then be semi-quantitative. Like many times when people start with these types of things, they start already with a goal that's semi-quantitative. maybe you don't want to say, you just want to say that this kid should get therapy and this kid should not. But if you start quantitative, you would know well, then it's easy to convert that into a semi-quantitative system. So the key would be, have you guys used Raspberry Pis before or PyCams?
I have, I don't know.
Is anybody in your team?
I think Matthew might have.
Yeah,
He's the one who does
between Kevin, Youngjin, and I mean, Raspberry Pi is just a computer, so you don't need to learn anything. PyCam is super easy. But get one kit, set up one of the kits in the area, get artificial blood from the lab, plenty in the lab. Just cheap blood would be an easy one.
Should I ask?
And that's started for
Also, you mentioned again, what's artificial blood?
Artificial blood is just kind of mock blood that's used for applications that early on when you're trying to test.
Like, is it a different animal or is it like a
There's
synthetic?
two versions. One is sheep blood, we have that too, but there's also artificial blood that matches the same viscosity and color, but is non-biological origin. You don't need any IRBs, anything for it.
It's safe to touch.
Yes, don't drink it. But also you should treat it like blood, wear gloves. There is the second part, which is sheep blood, which you should wear gloves, but it also has much relaxed IRBs. When it's human blood, it elevates. It's simple enough still to do. These are in vitro, so you have to do the bloodborne pathogen training. You should do that anyway. There is a STARS training for everyone. And since you are developing a test, you should know, because anybody doing this test would need to know those things. So you guys at the minimum should do the STARS training for the blood. Okay, let's keep going. Other teams. Who's next? Yes. Okay, so Mohammed, are you online? No. Okay, and then everybody else is in the class?
Yeah.
Yeah.
So we're still gonna meet to decide exactly something that's quick and easy to deploy for mosquito surveillance in areas that don't have systems set up already, because there's so many models currently that are used to predict like where the mosquitoes are going to move with climate change, but then yeah, you have to measure it, and also it doesn't mean much if there aren't any protocols for, okay, we predicted it's going to move there, what to do now. So we were thinking of a physical system that can do passive surveillance. I wanted to ask you, because you mentioned earlier in the class that you have done some things.
Yeah, have you guys read our 2017 paper? Okay, that's the first thing you
We
should
should
do.
do that.
We did this exactly, and that system has been scaling in a few places, especially Ethiopia and Puerto Rico. That's actually used currently. So it's a acoustics-based system. So any cell phone, you record 2 seconds of a buzz of a mosquito, and from the wing beat, we can actually tell what species. We built a ML model. Now that ML model was built in 2017, the world has changed. So the first thing that we could literally do is upgrade that model. We have the acoustics for 40 most dangerous mosquitoes in the world that we collected at CDC, traveling around the world. So all of that acoustic database is already there. And then it might be that we also know the challenges around acoustics. So then it implies. So for example, we build a lot of models that were around noise, ruffling that happens. People are talking in the background, recording these sets of things. So there's certain sets of scenarios. And in that, we can also go in other sets of directions. Around the same time, there was another class of papers where people were looking at lasers and a mosquito passing through a laser beam, getting the same information effectively, but it has less noise. But I think now I feel models have gotten so good that noise is not that big a deal. And then you're not building 5 billion of cell phones already exist in people's pockets. And so we built an entire citizen science program on top of it. So one would be is very specifically kind of expanding that, what has already been built. But many of the students graduated, but we still have a very large community that literally uses and develops that tool. The other one would be is because we understand the gaps in it, it's actually useful for specific sets of things to target. in that space, there is a question that still remains, which is, could you tell by some probe of a mosquito whether a mosquito is infectious or not? At that moment, if a mosquito that's flying around you, is it positive or dengue or not? And so for filariasis, we had shown that in the acoustic beat, there is enough change because it infects the flight muscles. And so we were able to tell for L3, L4, and L5 stage of the worm, we knew that this mosquito that's about to bite you. So that's also published. You guys should read 2 of those papers. And then let's do a separate sit down either today or tomorrow on kind of a deeper dive. I could tell you the key gaps. Access to mosquitoes. We have mosquitoes in our lab. I would have to train one of you to rear them. It does take a week to revive them. We have eggs sitting. You can also catch some mosquitoes. Who else is in your team? Is everybody here or are some folks also distributed? Do you know where? Which country? Okay, Morocco. Is that Mohammed is not here? Or no, Tunisia. Tunisia.
North Africa.
Yeah. Somebody should be catching mosquitoes ASAP.
Yeah.
You guys can catch. Do you guys know if there are mosquitoes around? Now is the season, right?
Yeah.
Okay, so go catch some local mosquitoes. I'll teach you guys how to ID them. It's very important to embody and understand what is difficult about a problem before you execute on something. And I remember the first task I had given one of my graduate students when we had just started the lab in 2011 is go around and catch mosquitoes in Palo Alto and in the lab in the Stanford area. And after 3 hours, the person came back with 1 mosquito. So it also is a challenge, but sometimes you just go to the right spot so you understand why that spot. But either dead or alive, bring some mosquitoes so that we can kind of, yes. And again, you know, it's useful because then you could evaluate and compare some of these things. We can grow culex. We won't grow Aedes aegypti. We have access for Aedes aegypti, but it requires a lot more safety, security, double locks, and all that. So although we have the eggs, we won't do that. But culex is a local species, so it's super easy to grow. It's super easy to order the eggs also. So the eggs arrive dry. You put them in dishes, and kind of it's useful to learn that process to a certain extent. If you're catching local mosquitoes, then it's better not to grow because you get the diversity. When the grown mosquitoes, you don't get the diversity, and the whole point is napping. Okay, so mosquitoes to start, diving into the set of papers. I'll give and share with you guys the acoustic data sets. It's also on Dryad. Any other urgent thing on mosquitoes?
Like the citizen science activity that people have to go out and like intention.
That's correct. So I think we tried 2 of them. One was we programmed cell phones to go inside CDC light traps. We also have light traps in the lab that I can give you guys. You can hang them outside. Light trap attracts mosquitoes. It's also a useful frugal science tool to look at because a light trap costs $1,000. It makes no sense. If as a citizen I want to attract and amplify a number of mosquitoes, I should be able to make light traps for cheap. But so you can record inside the light trap, then it becomes passive. I prefer the approach of engaging citizens because the things we were trying to do is change people's behavior and also engage in areas like inside our house. So you get much more spatially diverse information. Both of them are interesting. It's shocking to see how heterogeneous mosquitoes are in the distribution. So when you go to a village, you can tell explicitly a tiny spot where there will be high density of mosquitoes. And then you keep asking yourself, and then you realize, oh, the pigs were being stored there. And pigs make massive amount of CO2. And so that's really what the first spot that brought the mosquitoes there. And then they slowly start dispersing. So heterogeneity has really been a huge issue in surveillance because you report a number, but you miss out very much in the ocean, same issue. Heterogeneity of parameters in the ocean makes for that same challenge that we cannot then have a good predictable power. associated. So this is sort of an important thread around, and then you might say, oh, drones are an interesting way. But I haven't seen as yet a good drone-based mosquito platform. There are some cheap drones that are out there, but there's not a good. So drones is one thing I feel have the capacity, like a light trap combined with a drone, but they make so much air, no mosquito would ever be found close to it. Unless you flip this around and drones suck mosquitoes. Ooh, that's a cool idea. You see what I'm saying?
Starfish larva.
Because at some point of time, if air gets pushed, it has to go through. So
A fan.
yeah, but if on the other side of the fan had a filter, is a drone an interesting mosquito filtering device? Because then you just fly it around in the village, and at the end, and you do I had talked about in the early phase of what was the plankton recorder. What if we do a mosquito recorder that as the drone is flying around, there's a tape? So you know spatially when the splat happened. I mean, just so everybody's aware, in UK, there is a citizens program for mapping for cars that you submit. You put a sticky tape on your license plate, you drive for a month, and you submit the license plate sticky tape into, and then they genetically map and show the splats. And from that, they map biodiversity. Mosquitoes could also be used that way, but mosquitoes are hard to splat because the lighter the object, the air current will, but for larger insects, and this was the data that was used to show the decline in mosquito species. But I'm now thinking a plankton recorder with drones, because I think this was the one gap I would say in what we did is humans, even if with a lot of effort, light traps and a lot of effort, swarms, like mosquitoes are collective animals, so they form swarms. So if you're not close to the swarm, you say there is no mosquito. And these swarms move over time. So I couldn't.
What if you have like a suction component just beyond the disruption of air that the normal?
Yeah, but it could literally be the same thing, because for something to generate lift, you have to pump air that it has to suck. So it's sucking from the top and pushing down. And so you operate it in a
So we get like a low flying one.
low flying drone and suck and has filters.
You start low and then go up.
Yeah, play around with those things. I mean, it's an interesting idea. I'm not saying it's the only one we should do, but it maps to the challenge that has existed, is the heterogeneity never gets mounted. Like when you are on top of lakes, when you're top of like forests, it's harder. What if you just have a drone do a normal routine and you come back with its splat map, whatever that looks like, and then look from there. So I'm just expanding your list of ideas of things to try. Now we should really find what is the highest density of mosquitoes around the area. So useful to think of the and you said you have a drone. Okay, so see, now it's like you can actually put it to good use. Okay, to be continued. Who else is remaining? Anybody's team here that's missing? Any other team? I know that there are more presentations than that.
I can go.
Go ahead, Ashish.
Yeah. So I also
Can you say who
just,
all is in your team?
it's just me and a student from my lab.
Okay.
And
You want to
anyone
recruit
who,
somebody from
yes, I'll be
room?
happy to.
Yes. Shrish.
Yeah, if anyone wants to join, yeah.
Yeah, go ahead.
So yeah, before I talk about that, I've also worked with mosquitoes in the past, and just wanted to share some things. We built as a postdoc, I was part of a team. We built these robots that would sit in the field and attract mosquitoes with CO2. And the idea was to draw them in and give them a food bait, like a sugar meal. And as they would eat, they would drop the saliva, and then we could test the saliva for pathogens. That was the idea. It was called Smart Trap. Smart is acronym for something.
Yeah.
But yeah, it is a challenge, like to attract mosquitoes passively.
Yeah, and the heterogeneity is hard. Yeah.
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 Okay.
Yeah.
So I we are working on a project that proposes to do PCR without electricity. And I thought it would be a very good project for this class because the theme is frugal.
Yeah.
And the idea is to do
Yeah, and then just
PCR.
before you mention, Ashish, do you know about SnapDX, by the way?
Snap DX.
Yeah.
No? Remind
Okay,
me, please. No.
after I'll talk about it. I think I'll send you the first paper in that space, but it's useful for you to know. But go ahead.
Okay.
Yeah, I'll tell you
Okay.
about it.
I did a short presentation last time we were just doing presentation. I'm not sure if you saw the video.
Yeah, I haven't seen the presentation per se, but what I'll do is one thing that we can do is briefly at the end of this class, I was going to talk about diagnostics. So I'll cover a small piece of the SnapDX puzzle, but it's useful because it's built as a platform. It's useful if it's in any way aligned with what you're thinking about if you want to use that. But go ahead.
Yeah, so it's a very visual idea. That's why presentation is good, but I'll try to
Yeah, I
explain it
think
in
the big
words.
thing to do would be is I want to make sure that at least everybody has the immediate resources needed to initiate. And what we can do is let's do a team sit down between you, me, and anybody who joins the team itself.
I would love that. Yeah.
Do kind of because I'll take a look at I've done most of the deck, but I haven't finished the entire video yet.
Okay.
And then the other part of this would also be is it will, because you are at your lab, so it's super useful in terms of resources, I'm assuming.
Yeah.
You have everything that's needed.
Sure.
Right.
Yeah. So the idea, yeah, we have resources. I'm here to brainstorm.
Yes.
So the idea is that PCR, as you know, requires heating and cooling, so like 30 times, and that's a very power intensive process. And when we were doing this study last year, we interviewed like 140 clinicians, diagnosticians, parts of NSFI Corps, just a customer discovery what the main bottlenecks were during testing. During pandemics. And when you ask people in low resource settings, LMICs, they say that they don't have access to electricity. In fact, I found that 9% of the global population don't have reliable access to electricity, which was surprising to me. So we thought how we could do PCR without electricity. So the idea is that if you have a magnet and a like a metal which is ferromagnetic, which is hanging from a string like a pendulum.
Yeah,
it'll stick to the magnet, but if you have like a flame just beneath it, that flame will heat this.
I like where you're going.
So if you know this idea,
Curie temperature for the magnet.
This is called a Curie pendulum, and it will perpetually oscillate, and its temperature will go up and down as it detaches and attaches to the heat source. And if you monitor the temperature of the metal, it kind of mimics like a thermocycler.
Yeah.
But we've tried this in the lab with nickel because it's easily available. The Curie point of nickel is 350 degrees Celsius. So it does go to 350 exactly and then comes down.
Yeah.
So the idea is to make an alloy of metals that have Curie point of like 95. So that it can execute these thermocycles with just a flame. And a flame is like the oldest tool that we humans have. Like people knew how to make fire.
I think even the SnapDX, the thing we built is isothermal. And we also use just boiling water, and we use properties of boiling water for the isothermal threads. Super useful.
Is that lamp?
Yeah, that one is a lamp assay. Yeah.
Okay, awesome.
In this, so just as a comment around the current thread would be is the bottleneck right now just the alloy itself? Because I could imagine the alloy temperature could still be higher, but the reaction temperature can be different because you can play around with all kinds of thermal tricks.
So we explored some thermal tricks like putting an insulator and putting the tube on top of it.
Yeah.
But there's some challenges there. We can discuss more later.
Okay.
This
and
technical challenges.
I love the idea. It's so beautiful. It's elegant, and kind of it lights a flame. It's good. One thread to do would be is let's do a quick side discussion, and I'll cover some of the is there a target to use? And I'm assuming you already have some amount of supplies to test for a given target?
We have supplies to test for E. coli, SARS-CoV-2, those kind of pathogens.
Yeah.
So we have resources. This is a very new idea, just like 7-8 months ago. So it's in initial stages. But yeah, I would love to brainstorm and
why don't we
take
do
it
this?
in the right
Maybe right
direction.
after the chat, and then Shrishti, I don't know if this is the team that would be relevant for you to join. Let's talk right before. Anybody else who has not had the chance to talk about the project and in terms of resources, because I want to just make sure that some of the key resources are accessible. Folks online that have not as yet, has everybody committed to a project other than Shrishti?
I'm not committed to Shivam.
Vaibhav, is that you, Vaibhav, or somebody else?
That was Shivam.
Okay, Vaibhav, you're going to Nigeria. Did you tell everybody that you're going to Nigeria? I think you're committed. Okay.
I wasn't able to present it in the last class, but yeah, I wanted to pitch the cysto diagnosis project as well.
Yeah, you should recruit people too, like anybody who hasn't. So Vaibhav is from last year. He's continuing the thread. We had built an ML model for schistosomiasis on planktoscope, and now next month, end of June, he's going to Nigeria with Hope and Yuxing in the lab. She was also another Frugal Science student in the lab, and there we did some threads on Congo. And we are finally, after a year's time, which is actually pretty good, we're finally testing the idea in a field, in a clinical setting in Nigeria. So I hope you're ready. And anybody who wants to work on diagnostics, especially the ML part of the story, clinical validations, thinking a little bit about, this is implemented in schools. So this idea of combining diagnostic services in schools, because schools is where kids are, and for infectious diseases, it's actually very valuable, especially for very large numbers. So we're thinking a little bit about what does surveillance look like in schools, but as you can imagine, it's also a vulnerable population. So it's tricky. It has to be done with the right ethics. There's a lot of moving pieces of the puzzle. So folks that might be interested in that space, you know, you don't just have to think about purely the technical pushes that we will make. And then Vaibhav, you should still put together the same Fred Park, the same set of threads, and especially the current missing pieces of the puzzle, and record a quick 10-minute thing. And even if you didn't present last time, upload that. Where are the current, it's just still on YouTube, right? Not on Discord or anywhere. It's useful to chop them up. You know what I mean? Because people who are excited about specific projects might want, and especially if I start sending it, I got some messages for people who have been listening to our class passively, wanting to engage as mentors. So a particular choral group reached out, and other sets. Yeah. It's lots of people. It's useful, but what happened, it's really nice, is to just chop them up in some ways. Yeah. Okay. Shivam, anybody else who's not in a project yet? Everybody is? Oh, anybody who has not talked about the resources that you need to execute or clarity on what you need to do ASAP?
There's also the non-invasive imaging group.
Oh yeah.
Yes.
All of our team are in different places, so we're not sure how to start.
Yeah, that's exactly the point of this discussion. So the purpose of this would be is, in your analysis, what's the most urgent thing that you can put on your plate? And it's perfectly okay if it turns out that that urgent thing is computational.
Okay. We have two techniques we want to test, mechanical indentizer and electrical impedance spectroscopy. Those are two things we want to test.
Yeah, I mean, on the electrical impedance spectroscopy, if you guys are excited, I would just go for onco, because we have so much data. I literally have data from the field. We have all of the certain sets of things. And EM as a tool is remarkable in terms of thinking about simulations. And so if you guys as a team were heavy on the simulation side, there's lots of phantom models of skin and different cutaneous regions for skin that can be implemented. We have access to COMSOL in the lab, and we can give you guys access to that machine so that that simulation platform becomes valuable. It'll give us a sense of, it would be perfectly okay to push first on the simulation side of the story. Also, building impedance spectroscopy is fairly straightforward.
Yeah.
So there is actually a portable impedance spectroscopy setup. And the one that I used is I hacked something that's used in, you know, when your internet is not working and you invite these people to come, how do they know where the line is broken? And so there is an impedance spectroscopy. It's a portable tool, and I hacked that to use in the field to get all of the spectras. And so that by itself is very widely available now because, you know, internet problems are a huge issue. And so we can start with that as a platform itself. And it's super easy to rent because it's a common tool that's given out for $50 or $100 a week. We can even rent something like that.
Okay.
So Indumita, who else was in your team? Is everybody online?
Yeah, it's Kamal, Kershishtik, and then there's one more person, but
I see.
Manan, but I'm not sure
Yeah.
who's.
Why don't we do this? Let's have a brief chat, but try diving in to see on the simulation sides what you need, and look in the commercial space of this, the cable testing capabilities, what the overlaps actually look like. And I will share a presentation with you guys on the data on the Onco, and then we'll build from there.
Would a voltmeter also work as checking?
Yeah, no, it's not. You have to have both a source and a receiver. So it's basically a broad frequency source, and it measures at the same time, and it's quite sensitive. But what's used is, it's essentially either a 2-point or a 4-point measurement, but it's extremely, the cost logistics have been such that because it's so commonly used, it's now become low cost. Okay, anybody else? If your project hasn't been discussed, and oh, Shrishti, go ahead.
So it only exists in my imagination. I was wondering if it's possible to build like an outdoor air purifier, because I am currently located in Delhi NCR and the air quality is really bad. And I was wondering if we could create a network of outdoor air purifiers, and that would like collect all the pollutants from the air, and we can like classify them and reuse some of the pollutants if it's possible, and eliminate the ones that are like bacterial or some kind of causing some kind of sickness. And if we try to build this,
Let me
will it
just
be a very
say, I think
far-fetched goal?
now it would be, I think putting a completely new idea at this moment would be too late, unless there was a
Yeah.
team already that was working on air quality. And we discussed air quality before, but I don't think any current team is working on it, right? Because I want to make sure that people are not working alone. Working alone is not
Yeah.
because of the class. You can file ideas that you're excited about for later, but try working in teams of 3 or 4 people, because that's really when you can make progress. And it's not about, it's about choosing, it's totally independent of what you end up choosing to work on. What's most important is executing with a systematic approach to it. So that's far more important, I would say. So although I'm super excited about air quality, we can talk at length about it. There's a lot of citizen science projects on using full scope to measure the carbon soot levels in the community. There are automated versions of it too. But unless you can find 2 more people in the team to work on air quality, I would say punt it for right now. And then I would say to join a team that's already functional and has kind of anchored in on what they want to do. And again, the purpose of this is practice, so that you're not operating alone. Anybody else who is not part of a team? Because otherwise I'll call upon you guys at some point of time.
Hi, Manu.
Yes.
I'm not part of
Okay.
any of the team, but you were just recently talking about one of the teams who's working on mosquitoes.
Yes.
So, yeah, so over the last couple of weeks, I've been working with this PhD student in doing a taxonomic classification of a lot of mosquitoes that they collected from Western Ghats in India. So
Awesome.
so we identified a lot of mosquitoes, including Aedes, Anopheles, Autopodemia, Hoatzia, and
Yeah.
stuff.
Okay, so here's a person who has some taxonomy knowledge. Okay, so join the team. And where are
Cool,
you
yeah.
based in, Tushar, right now?
Ashoka.
Fantastic. Okay, and Sonipat has a whole bunch of mosquitoes, right?
Yeah.
Okay, catch some mosquitoes. All
Cool,
right,
yeah.
recruited in the mosquito team.
Okay.
Just Discord, yeah. You guys figured that. I don't know. Nobody's using Discord. It feels very quiet there. Whenever I go, no?
Using WhatsApp.
Okay. Yeah. If you're using WhatsApp, is there a way to do a WhatsApp group? I mean, I'm happy to move everything from Discord to WhatsApp, like a class group, which has what I want to see is chit chat on every project. So I can just chime in and say, oh, that's the point, because otherwise it sort of misses out. Then it only happens here, but it's useful. Whatever you guys are using, just include me. There's no oversight. That's the whole point, is undisciplined. Yeah. Yeah. But also what I'm saying is there is a second feature I learned in WhatsApp is to make WhatsApp community. And then WhatsApp community can have multiple channels that then, so this is the Nigeria project that's happening currently. This is the community page. And then every one of them is a sub page, which is a WhatsApp community. So it would be useful if people are using WhatsApp, we can transition from Discord. It's just, Discord has a gaming feel to it. And I mean, I can understand. I don't like it either. Okay, anybody else? I think the biggest thing I want to just make sure is people have resources. And if you don't know where to start, please talk to me. I'm going to transition to talking to individual teams now. And then maybe we will do the Wednesday morning, let's do the diagnostics lecture so that we can cover the breadth of diagnostics. I'll also bring some materials. But ensure if you want to sit down with me now, I still have another half an hour or so. So I'll sit down with the plant team. Anybody else? Yes. So let's do that. Why don't we do this quickly and then that, is whatever order that works. Yeah. Anybody online that wants to sit down and chat?
Actually, Manu, can I also join in for 5 minutes?
Okay. You want to talk after? Right. So just stay online. So I have 3 groups right now. And then Ashish, I don't know if you have time today or do you want to talk a little bit later?
Do you have time today? Because you 30 minutes.
Yeah, I think 30 minutes
groups
are gonna fill up and then I have to run.
We
But
can do it
why don't
next
we
time.
do tomorrow is actually ideal.
Yeah, that's fine with me. Yeah.
Why don't we do a separate chat? And we can do a
I'm in
slightly longer chat.
I'm in India right now, so I'm on Indian time, but I can
I see. I didn't realize that. Oh my goodness. Yes.
So yeah, it's fine.
Okay.
So do I join Ashish Priya's team now?
Yeah, you can join my team. I can
Yes.
brief you on the project and
Why don't you guys
meet
talk
with
separately?
Manu. We can meet with Manu
Okay.
tomorrow. I'll set up a
Yes.
meeting and sounds good.
Okay, so will you ping me on Discord about it?
You're on Discord, right? I'll find you.
Yes. Okay.
Yeah.
Okay. Thank you so much.
All right. Sounds good. I'm going to jump in with, let's do the coral thread. Let's do plants. And then we will do web of. Anybody else?
Manu, are you talking to the mosquito team as well?
The mosquito team, do you guys need me today? You want to talk today or later? I think, no, mosquito team should read what we did first. You guys should, let's do tomorrow. Yeah. Tomorrow morning, essentially. Yeah. So the mosquito team, Tushar, you should just first talk to the mosquito team and then we'll follow up.
Okay, yeah, good.
Yeah. Okay, all right. I'm gonna keep this running for a little bit, or
I need to adapt.
okay. Webhub, we'll talk on WhatsApp. We'll do a WhatsApp video. Yeah.
Right.
Bye, everyone.
Professor?
Oh,
Okay,
go ahead.
sorry.
Shivam.
Yeah, which team do I join?
Oh, you
I'm a
haven't
bit
joined the
confused.
team. I was.
Yep. I
Uh.
was about paper, but I think I spoke to professor and they have not published the data. So yeah. So I missed on that project because I think that might take some time to
No,
publish and
that's
all. So
fine.
therefore
Just
I had to.
for all the projects that are happening, have you watched all the videos?
Yes, I was there in the last class, so I've seen
Okay,
it.
so then what's the
Yeah.
project you're most excited about?
I have 2, 3, one the Ashish Priya one, and I think now even the one which is by Vaibhav and the Tushar. So Tushar and Vaibhav are on campus and we meet.
I would
So
say
I think it
join
will be easier.
Webhub. Yeah, join Webhub. It's useful if you're local there. And Webhub has a lot to do before we ship him to Nigeria. So he can use some help. And
Sure,
he
sure.
also
Then I'll
has a
join them.
microscope, so you kind of get a chance to sort of build it. There's a lot to be done itself. I think it's,
All right.
although I like the idea of cross teams, there is also usefulness in sitting together.
But I just don't know if like in what ways will I be able to contribute because yeah.
There's an
So
infinite amount of work to be done.
okay, all right.
Yeah,
Sure, sure. Then I'll
right.
join.
Yeah,
Thank
you
you.
should join, like quite literally. There's never, you know, you have to remember when you're trying to solve a problem, it's not what you know, or you have to ask yourself what needs to be done and then learning to just execute on that. Because then, especially when you put some goalposts like, oh, we're doing a clinical validation study for something like this, suddenly the clock is ticking because samples are going to arrive there, or you're doing something with corals. The corals are there. They don't care about whether you are ready or not. They're going to come and they'll die. So similarly, samples will come and they'll disintegrate. And so working backward from it, and I think improving the models is currently the best, and testing some amount of that testing beforehand for what are the false positives currently, and understanding those false positives and saying, oh, should we be filtering some of the urine to remove false positives? Because when people pee, they also pee blood, they also pee all kinds of things because these are sick kids. So we have always tested it with healthy pee. So the biggest thing I am worried about is there's going to be too much blood, there's going to be too much dead lysis cells. So are the models actually capable of handling that? So think a little bit about what does that mean and how can we filter that out? Because if you try filtering that out, you actually skip the eggs. And so building a little bit of resilience in the current models is actually critical.
Mhm.
Okay, to be
Right.
continued. I'm going to hang up here because Kevin has to run, and Weber, ping me on WhatsApp, and Shivam can join too. Bye, everyone.